Hello students, welcome to EPG Pathshala. I am Dr. Sushma Talegaonkar from Jamia Hamdard. Today we are going to discuss about the characterization of multiple emulsion system, what are the various stabilization processes in multiple emulsion system and application of this multiple emulsion system in pharmaceutical field, especially in drug delivery under the paper Novel Drug Delivery System 2. The learning objectives of the today's module, they are: first, we will discuss in vitro and in vivo characterization of multiple emulsions. Then second is factors which affect the stability of multiple emulsion system. The third one is what are the various approaches to improve the stability of multiple emulsion system and applications of multiple emulsion system. First, we are going to take the characterization of multiple emulsion system. Under the characterization, we divide this characterization parameter especially into two parameters. First, we are going to discuss about the in vitro characterization, and the second one is in vivo characterization. The characterization parameters are very important in especially in any formulation of pharmaceutical preparation, because until unless we are not going to characterize, we cannot determine the stability and the fate of particular formulation in biological system. So under the in vitro characterization, we are going to discuss about the various parameters like what is the size and size distribution of the dispersed globules in multiple emulsion system, what is the surface area of multiple emulsion system, what is the zeta potential of multiple emulsion system, because zeta potential also play a very important role in stability, and by taking the zeta potential reading, we can also determine the stability of multiple emulsion system. Then we will also discuss about what is the entrapment of the drug in multiple emulsion system and what is the release pattern of the drug from multiple emulsion system. Under this characterization, we are also going to discuss what are the various novel methods the scientists they have designed to optimize and to determine the each and every parameter. In vivo characterization, we will discuss about what is the fate of multiple emulsion system in biological system, what are the various routes of administration through which the multiple emulsion system can be administered into the animal body or the human body, and what are the biodistribution studies of the multiple emulsion system, means when we are giving the multiple emulsion system by oral route, how it is going to be distributed. When this multiple emulsion system, when we are administering through intravenous route, what will be the distribution pattern, whether the drug is generally is going to be released in which part of the body, and also in case of multiple emulsion system, when it is given by a topical route, what will be the fate, whether it is generally breaking this skin hurdle for the absorption, and how this multiple emulsion is going to release the medicament through the skin, and the drug is going to be enter into the blood. So there are the various uh, applications and the characterization parameters which are very important in designing of stable multiple emulsion system. In vitro characterization of multiple emulsion, the first characterization parameter is average globule size and size distribution. The optical microscope with calibrated ocular and stage micrometer can be used to determine globule size of both internal dispersed phase droplets and multiple emulsion droplets. Other than optical microscopy, techniques such as scanning electron microscopy, counter, freeze fractured electron microscopy, and recently introduced NMR self-diffusion methods are used to characterize the multiple emulsion system. Second, area of interface. The following formula is used to calculate the area, that is S is equal to 6 upon D, where the S is equal to the total surface area of interface in square centimeter, D is diameter of the globules in centimeter. Third parameter is rheology. The rheology of multiple emulsion is a significant parameter which relates to emulsion stability and clinical efficacy. The two major parameters, viscosity and interfacial elasticity, are used to determine the product rheology. First, viscosity or viscosity changes. It is generally measured by Brookfield rotational viscometer or rheometer can also be used. Second, the determination of interfacial film strength. The film strength of single and mixed emulsifier systems characteristically used in multiple emulsion formulation is determined by interfacial elasticity measurement using an oscillatory ring surface rheometer at oil-water interface. Conductimetric analysis. The specific conductivity of the multiple emulsions is measured directly using a digital conductivity meter. Drug-loaded and unloaded emulsions can be differentiated by measuring the conductivity values, which generally increases in drug-loaded formulations. Another parameter is pH determination. The pH of fresh multiple emulsion and emulsions kept in different storage conditions is determined by digital pH meter in order to determine any chemical degradation occurred in different storage condition. Next, centrifugation and phase separation. Emulsions are centrifuged at 25 degrees centigrade and this test is performed at the different storage condition at a specific intervals. The result of centrifugation indicates the stability or instability of emulsion against the phase separation. The phase separation can be calculated by the formula, that is percentage phase separation is equal to the amount of the phase separated divided by the total volume of multiple emulsion multiplied by 100. Next is zeta potential. Since the electric charges on the particles affect the rate of flocculation, zeta potential is an important parameter used to determine the emulsion stability. The zeta potential measurements are essential to design the surface modified or ligand attached functionalized multiple emulsion system. The zeta potential and the surface charge can be calculated from the mobility and electrophoretic velocity of the dispersed globules using zeta potentiometer by the help of following equation. Z is equal to 4 pi eta micron upon eta e into 10 raised to power 3, where Z is the zeta potential in millivolt, eta is equal to the viscosity of dispersion medium in poise, micron migration velocity in centimeter per second of dispersed globules, eta dielectric constant of the dispersion medium, and E is the potential gradient of the system. Next is the percentage drug entrapment. Percentage drug entrapment in multiple emulsion system is generally determined by dialysis method, centrifugation method, filtration and conductivity measurements. Evaluation of drug entrapment is made by using impermeable internal tracer or marker molecules contained in the inner aqueous phase of multiple emulsion. The unentrapped marker is calculated and the amount entrapped can thus be calculated by subtracting unentrapped amount from initially added amount. Next is in vitro drug release. This is again a very important parameter to decide what is the release pattern, whether the multiple emulsion is sustained release or immediate release. The drug release from inner phase of a multiple emulsion can be determined by conventional dialysis method. The emulsion is placed in the dialysis bag and dialyzed against the saline phosphate buffer pH 7.4 at 37 plus minus 1 degree centigrade, and a sink condition is maintained during the process while sink contents were stirred continuously using a magnetic stirrer. Cumulative drug release profile was calculated by withdrawing the aliquots at different time intervals and assessed using standard procedure. The graph is usually plot between time intervals against the drug concentration taken on y-axis, and this graph is showing the sustained behavior of multiple emulsion system. In vivo characterization of multiple emulsions, Under this heading, the following parameters are generally determined. First, pharmacokinetic study, pharmacodynamic study, and biochemical estimation. Pharmacokinetic studies. The pharmacokinetic studies are generally performed to determine the drug concentration in the blood or the plasma to indicate the release of the drug substances from the multiple emulsion into the systemic circulation. In this type of the study, various pharmacokinetic parameters such as the rate of clearance, volume of distribution, and absorption and blood plasma levels are generally determined by a suitable analytical methods like HPLC, LCMS, etc. Pharmacodynamic studies. Pharmacodynamic studies, they are generally performed in disease-induced model in which we are generally observing certain parameters in diseased model of the rats or the mice or the guinea pig, and we are generally evaluate the certain parameters by injecting the multiple emulsion formulation. Biochemical studies. In these studies, various disease-related biochemical parameters are determined by taking the blood or plasma or the serum or the urine samples using the different methods such as enzyme-linked immunoadsorbent assay, Western blot technique, etc. Now let us see what are the factors which affecting the stability of multiple emulsion system. There are the various factors which are generally considered very important to affect to the stability of multiple emulsion system. They are first nature of entrapped material, shear or agitation, temperature, rheology or the viscosity of the multiple emulsion, and aided stabilizing components in multiple emulsion formulation. Let us discuss one by one. First, nature of entrapped material. The material which we are going to incorporate in the internal phase means in case of water in oil in water emulsion, the internal aqueous phase, or in case of oil in water in oil emulsion, the internal oil phase. It will generally play a very important role in stability of multiple emulsion system, and they are generally the nature of the internal material will generally maybe hydrophilic or hydrophobic, and it is generally make a great impact on the stability of multiple emulsion system. So sometimes we generally, to enhance the stability of multiple emulsion system, we usually put some osmotic agent in internal aqueous phase like sodium chloride and mannitol. They are very popular example which we are generally incorporate in internal aqueous phase. So they generally maintain the balance between the uh, internal aqueous phase and outer external phase, and they are generally prevent the coalescence among the droplets. Second important point is shear or agitation. It is again a very important parameter which generally govern the stability of multiple emulsion system. So high shear may be cause the disruption of large percentage of multiple oil droplets, which result in increasing the surface area and unstable the emulsion formulation. And that's why we have already discussed in during the discussion of preparation of multiple emulsion system that in case of first step emulsification, we usually generally accept this high shear equipments, because primary emulsion, they should be very stable, and if we are using the high shear for their preparations, they generally lead to the stable primary emulsion, and if the primary emulsion will be stable, definitely they will provide a stable secondary emulsion. But if we are using this high shear equipments in second step of emulsification, then ultimately it will lead to the breakage of emulsions and the leakage of the drug molecules, and total purpose of multiple emulsion is going to be failed. So one should be very careful when he or she is using any shear equipments for the preparation of multiple emulsion system. Let us take another parameter which can affect the stability of multiple emulsion system, that is the temperature. The temperature plays a very important role, because this temperature will affect the solubility of this surface active agent in dispersion media. And this temperature will also affect the viscosity of the multiple emulsion system. So in literature, they have reported that this temperature is generally having an indirect effect on multiple emulsion system. But one should be very careful regarding the temperature during the preparation of multiple emulsion system. Another important parameter, that is the rheology or the viscosity of multiple emulsion system. Generally, the water in oil emulsions, they are having a low viscosity as compared to oil in water in oil multiple emulsion system. And that's why if we will see the stability point of view, the oil in water in oil emulsions, they are stable if we are talking about in terms of physical stability. So they are more stable as compared to water in oil and water emulsion system. But at the same time, the oil in water in oil multiple emulsion system, they are very prone towards the rancidification or oxidation, and they are ultimately lead to a very bad smell to the system. While in case of water in oil in water emulsion system, their viscosity is quite low as compared to O by W by O. And many a times we generally incorporate a viscosity enhancing agent in order to enhance the stability of water in oil in water system. This viscosifying agent we can add either in internal aqueous phase or external aqueous phase. And due to this enhanced viscosity, their coalescence will be less and stability of the multiple emulsion will be increased. There are so many papers which are generally reporting the use of gelatin or the polyacrylic acid in internal or external aqueous phase to enhance the viscosity of multiple emulsion system. Approaches to improve stability of multiple emulsion system. The first approach is gelation of phases by gelation of internal or external phase or by gelation of oil phase. The gelation of either internal or external phase or oily phase has been employed in various studies to stabilize the system. The stable water in water emulsion have been formed by polymeric gel in internal or external aqueous phase. Example, polyacrylamide gel in oil in water system or water in oil in polyacrylamide gel system. The second approach is the formation of interfacial complex film by in situ polymerization at the interface or by interfacial interaction between a polymer and the surfactant. In this method, we generally this mode of stabilization can be executed either through in situ polymerization at the interface or by interfacial interaction between a polymer and a surfactant. Interfacial complexation between non-ionic surfactant and macromolecules has been employed for stability. Another approach to obtain the stable water in oil in water multiple emulsion is based on interfacial interaction between macromolecules. Example, albumin or bovine serum albumin or polyacrylic acid or globulin, they contain in the internal aqueous phase. A lipophilic surfactant or high molecular weight polaxomer 331 was preferable in formation of stable water in oil emulsion and subsequent water in oil in water emulsion. Similarly, the hydrophilic polaxomer surfactant of high molecular weight like polaxomer 403 was found to enhance the stability of water in oil in water emulsion when used as a emulsifier. The third approach is modulating the surfactant concentration. A stable emulsion is formed when suitable surfactant with required HLB is used. Stability of emulsion has also been enhanced by using the mixed emulsifier, as this tends to form a more stable interfacial film. Another approach for the stabilization of multiple emulsion is presence of additives in internal aqueous phase. Electrolytes have been used for obtaining a stable water in oil in water emulsion. These electrolytes include the ascorbic acid, acetic acid, sodium chloride, and sodium citrate. The addition of lysozyme to the aqueous phase or sterile amine or oleic acid to the oil phase effectively improves the stability of oil layer. The influence of the various additives, example glucose, sucrose, acetic acid, citric acid, ascorbic acid, and sodium acetate on the stability of multiple emulsion have been studied, and it was concluded that glucose and the sucrose could increase the viscosity and as a result improve the stability of emulsion system and by decreasing the coalescence, and they also help to maintain the tonicity or the osmotic pressure between the water internal water phase and the external water phase, and hence the to prevent the leaching or the coalescence of the droplets. The another approach for the stabilization of this multiple emulsion is pro-multiple emulsion formation. In this approach, the emulsion is stabilized by changing the physical nature of emulsion from liquid to solid by using the adsorbent or the solids. Whenever the multiple emulsion is needed, this pro-emulsion is dispersed in the specified quantity of water to form the multiple emulsion. This is a new approach and is still under the extensive investigation. But if one will get the success in this approach, definitely the commercialization of the multiple emulsion is possible. The next and the last approach for the stabilization is a steric stabilization of multiple emulsion. In this approach, the steric stabilization can be achieved by using either the adsorption or grafting of the natural polymer or by using the polymeric surfactant or lipid grafted polymers. This steric cloud may help to prevent their instability due to the environmental challenges. The multiple emulsion can be stabilized by coating them with the hydrophobized polysaccharides like amylopectin or poloxamers or the pluronics. Applications of multiple emulsion system. Multiple emulsions are used in different areas of therapeutics, which are given below. First, control and sustain drug delivery. The release of the drug from the multiple emulsion can be prolonged or controlled by incorporating the drug in the innermost phase of multiple emulsion. Because a drug has to pass through the several phases before it is available for the absorption for the system. Most of the water soluble drugs are used as multiple emulsions because it has become possible to control the release rate of medicine and suppress the strong side effects as compared to the simple emulsion. Water in oil type simple emulsion generally has such a high viscosity that create the difficulty in infusion to the arteries or capillaries via catheter. Also, the O by W emulsions are not an option because they do not encapsulate the drug. But water in oil in water emulsion systems are suitable drug carrier because they have the capability to encapsulate the large number of the drug in internal aqueous phase and produce the low viscosity due to the external water phase. For the application of water in oil water emulsion as a drug delivery system, it is important to prepare a very stable water in oil in water emulsion in which the countless submicron water droplets are encapsulated. Prolonged release of multiple emulsion of cis-mercaptopurine were prepared by Khopre et al. By the incorporation of sphingomyelin and monocyloganglioside GM1 in the oily phase and by coating with the lipid grafted polyethylene glycol. It was concluded that PEG-PC coated multiple emulsions are superior as prolonged release and extended the blood circulating carriers compared to the multiple emulsion having the GM1 or SM. Another example in which the anti-diabetic solid in oil in water, that is S by O by W emulsion, was developed for oral administration of insulin. Surfactant coated insulin was dispersed in the oil phase by ultrasonication, and this dispersion was mixed with the outer water phase with a homogenizer, and finally the sphere in oil in water emulsion was obtained. The sphere in oil in water emulsion shown hypoglycemic activity for a prolonged period of time after the administration to the rats. Second application, the multiple emulsion can be used as an oxygen substitute. Hemoglobin is generally as a fragile material is formulated as a water in oil in water multiple emulsion system in physiologically compatible oil in an outer aqueous saline solution. It has sufficiently small droplet size to provide the oxygen flow through the blood vessels to the desired body tissues or organs, and thus providing a blood substitute for oxygen delivery. This type of multiple emulsion contain high hemoglobin while maintaining the high yield and high oxygen exchange activity. The third application of multiple emulsion is a bioavailability enhancer. The multiple emulsion system can be used as a bioavailability enhancer. They increases the bioavailability of various drugs either by protecting the drug in physiological or enzymatic environment in GI tract, where otherwise these drugs get degraded like protein peptides, and they can also protect the drug by hepatic first pass metabolism. Multiple emulsions are used to enhance the bioavailability and also prolong the drug delivery of various compounds in biological system. Multiple emulsions have been used to improve the bioavailability of various lipophilic drugs such as the griseofulvin, nitrofurantoin, atorvastatin, lamotrigine, rifampicin, diclofenac, sodium, which have a high first pass metabolism. Taking an example of multiple emulsion of atorvastatin, which has been prepared by Vyas et al. By two-step emulsification process using non-ionic surfactant, and they found the controlled release of the drug as compared to tablet. The next application of multiple emulsion is a targeted drug delivery system. Multiple emulsion have shown the great potential for drug targeting to the lymphatic tissues because of the uptake by the reticuloendothelial system. Therefore, it acts as a lymphotropic drug carrier to several organs such as the liver and brain. Multiple emulsion had also been prepared for several cytotoxic agents targeting different tumors like bleomycin and paclitaxel, etc. These can be used as a lymphotropic carrier for drug targeting. A stable multiple emulsion for lung targeting containing rifampicin was prepared by Khopde et al. In the internal aqueous phase was prepared by incorporation of additives in both the aqueous and oily phase. The in vivo studies performed with stable multiple emulsions administered orally in human volunteers showed prolonged plasma drug levels and were found suitable for an improved tuberculosis therapy. Inverse targeting. In this approach, the surface modification provide the stealth property to multiple emulsion carrier system. Regarding this approach, Talegaonkar and Vyas prepared a sphere in oil and water, that is S by O by W type of multiple emulsion of diclofenac sodium containing poloxamer 403 coating. They gelatinized the inner aqueous phase and studied the effect of poloxamer 403 coating on the surface of the multiple emulsion for inverse targeting to reticuloendothelial system rich organ. The results of this study concluded that the multiple emulsion system containing the poloxamer has a capability to retard or to bypass the RES uptake of drug or multiple emulsion mainly to liver and spleen, and targeting to non-RES system such as the inflammatory tissues, lungs are possible. Another important application of multiple emulsion is taste masking. The taste of certain bitter drugs can be masked by placing the drug in innermost phase of multiple emulsion. Multiple emulsion of chloroquine, an antimalarial agent, has been successfully prepared and had been found to mask the bitter taste efficiently. The taste masking of chlorpromazine, an antipsychotic drug, has also been reported by multiple emulsion system. Next application is treatment of drug overdose. water in oil in water system with aqueous base in oil was designed to remove the acidic drugs like barbiturate and salicylates from gastrointestinal tract by entrapping the unionized drug permeating through the oil membrane into the inner basic phase, where it is converted to an oil insoluble anion. This system could be utilized for overdoses treatment by utilizing the difference in the pH. In this emulsion, the inner aqueous phase of emulsion has the basic buffer, and when emulsion is taken orally, acidic pH of the stomach acts as an external aqueous phase. In the acidic phase, barbiturate remains in unionized form, which transfers through the oil membrane into the inner aqueous phase and gets ionized. Ionized drug has less affinity to cross the oil membrane, thereby getting entrapped. Thus, entrapping the excess drug in multiple emulsion cures overdoses or drug poisoning. Another application is enzyme immobilization. Most enzymes in industrial and pharmaceutical processes are relatively unstable, and the cost of their production and purification are still very high. Therefore, enzyme immobilization on various carriers have been developed. Enzyme immobilization via multiple emulsion technique involves the entrapment of enzyme which catalyzes the process in the interior phase of emulsion. Alcohol dehydrogenase has been immobilized by multiple emulsion method and used in conversion of alcohol to aldehyde. Highly lipophilic substrate such as steroids can be carried out in a multiple emulsion. The enzyme is contained in a microdroplet water pool, whereas the organic phase contains the substrate solution. For example, hydrocarbon-based liquid surfactant membrane have been used to immobilize urease enzyme. Next application is topical application. Renault and co-worker in 1993 developed a topical water in oil in water emulsion containing sodium lactate in inner phase, spironolactone in the intermediate phase, and chlorohexidine gluconate in outer phase. Other studies entailed comparison in the release profile of metronidazole from water in oil in water, and water in oil in system by Ferida et al. And Ozer et al. Few studies have been made on the topical administration of water in oil in water multiple emulsion. Although there are advantages in the controlled release by oral and parenteral administration have already been shown with metronidazole, caffeine, and dihydralazine. Next application is microencapsulation of drugs. The two-step emulsification procedure of preparation of multiple emulsion is very popular in preparation of solid microsphere or microcapsules. The difference is only in place of oil we are using the organic solvent. So we can say the water in organic phase in water method is generally used to prepare the microsphere dosage form. Among the microencapsulation technique, this multiple emulsion solvent evaporation is one of the most useful method for entrapping the water-soluble compounds, generally the protein and peptides and some enzymes. Drugs of various therapeutic category like theophylline, salbutamol sulfate, diclofenac sodium have been encapsulated using this technique. Lee et al. Was prepared floating polymeric microcapsule that simultaneously entrap the multiple drugs were prepared using a solid water uh, in oil in water emulsion solvent evaporation method based on interfacial phenomena and manipulation of solvent removal process. The fabricated microcapsules exhibited the excellent buoyancy in simulated gastric fluid and provided the control and sustained release of multiple drugs for up to 24 hours, and thus revealing their potential as a rate controlled oral drug delivery system. Another application of multiple emulsion is vaccine or vaccine adjuvants. Herbert reported the use of water in oil in water multiple emulsion as a new form of adjuvant for antigen and found that these emulsions elicited the better immune response than antigen alone. Rishendra and Jaiswal developed a multiple emulsion vaccine against the Posterula uh, multocida infection in cattle. This vaccine contributed both the humoral as well as the cell mediated immune responses in protection against the infection. It was concluded that this multiple emulsion based vaccine can be successfully used in effective control of hemorrhagic septicemia. Another application of multiple emulsion is in food supplements or food application. Double emulsion is generally used in food industry. Preliminary study have been performed in the field of entrapment of flavor components in a release system. Sensitive food material and the flavoring agents can be encapsulated in water in oil in water emulsion system. Sensory tests have indicated that there is a significant taste difference between the water in water emulsion and oil in water emulsion containing the same ingredient, and that there is a delayed release of flavoring substances in double emulsion system, and the effect will be last or last longing, or we can say for a prolonged period of time. So, student, let us see what are the conclusions which we can make from the today's module. The first conclusion that the multiple emulsion system, they are the complex system, and they are generally characterized for various in vitro and in vivo parameters. If anybody wants to prepare a stable multiple emulsion system, he or she should characterize the multiple emulsion system thoroughly, then only he or she can achieve the stable multiple emulsion system. The multiple emulsions, they are generally stabilized by various techniques, and one should be very careful in optimizing the technique for the stabilization. And the substances which we are generally use for their stabilization, their physical, chemical and physiological consideration should take in mind, so they should not make any incompatibility during the formulation. Application, if we will see the application, the multiple emulsion has a wide variety of applications, but among these applications, the sustained drug release or controlled drug delivery is one of the most important application in pharmaceutical field. And due to their instability, they are not that much popular, but if we will see their applications, and if anybody will try to stabilize them by making the concept of pro multiple emulsion, the industrialization or commercialization is not a very difficult task. And if we are selecting a proper stabilizing technique, one can achieve a stable multiple emulsion system. Thank you.